Good morning, biology students. Today we are going to be continuing with chapter 7, which is cells. So when we left off yesterday, we were talking about two of the domains of life, the prokaryotes and the eukaryotes. So prokaryotes are generally small and simple. They are much smaller and simpler than eukaryotic cells. The organisms we call bacteria tend to be prokaryotes. Prokaryotes are quite simple. They have DNA like all cells. Their DNA is going to be in some cytoplasm. They have some ribosomes. They have a cell membrane. They have a cell wall. They can have a capsule, which is like a second cell wall. They can have pyluses to have them grab onto things. They can have flagella for swimming. But that's really all their parts. In contrast, eukaryotic cells are generally much larger, much more complex. Most of today we are going to be talking about the organelles of eukaryotic cells. So we're going to be talking about the organelles that we find in animal and plant cells and fungi. So if you look up here, all of these tiny little blue dots are bacteria. For comparison, the big blue dots in these cells are cell nuclei. So the bacteria are much smaller. Eukaryotes are much more complex. So the first distinction that we're going to be talking about today is between the cytoplasm and the nucleus. So everything that is outside of the nucleus, which is the large structure that holds the DNA, can be called the cytoplasm. Both prokaryotes and eukaryotes have cytoplasm. It is the fluid portion of the cell. So inside of cells, they have structures that we call organelles. Organelle literally means little organ. So these are specialized structures that do specific jobs. Most of the rest of these notes are going to be going over the jobs of these different organelles. When you are back in the classroom, we are going to be doing a lab. We're going to do a plant cell lab where we can see a few structures, so you should get to look forward to that the week that you are back. So the first structure we're going to talk about today is the nucleus. It is the largest, most easily identifiable structure in a eukaryotic cell. The nucleus is a control center. It contains nearly all of the DNA. The nucleus is surrounded by a nuclear envelope, which is made of two membranes. So just like your cells have a membrane, the nucleus has a membrane. It actually has a double membrane. So that nuclear membrane is covered with nuclear pores. It doesn't have just a few dozen like this picture is showing. It would have thousands. Those nuclear pores allow material to get into and out of the nucleus. The nucleolus has a dense region within it. The nucleus, rather, has a dense region within it called the nucleolus. The nucleolus is where ribosomes are assembled. So ribosomes are another organelle. They are responsible for assembling proteins from their amino acids. So next organelle is a vacuole. A vacuole is basically just a big bag. Vacuoles are large sac-like membranes that store materials like water, salts, proteins, and carbohydrates. They tend to be a lot larger in plants than in animals because plants can't walk around to get a drink. They have to store water, whereas animals generally aren't storing as much water as a plant would. So plants have a large central vacuole that keeps plant cells rigid. This is what keeps vegetables crisp. Once a flower has started to wilt, its central vacuole has lost enough water where it can't support itself outwards. We also find vacuoles in other organisms like animals. Some single-celled organisms have what we call a contractile vacuole, and these contractile vacuoles can allow them to move around. So This paramecium can fill water up into a vacuole, and in any second now, he's filling, and then he can start to zoom away by squirting the water out. So a somewhat similar organelle to a vacuole is a vesicle. Vesicles are used to store and move materials between cell organelles as well as to the cell surface. So vesicles are made of membrane. They're made of the same stuff as the outside of the cell. What's convenient about that is they can just join up to the outside of the cell and dump stuff out, or one can be made where it just sort of pinches around stuff and pulls it in. So these vesicles can be used to move things around a cell. So that big blue blob is a vesicle that is getting moved by a motor protein, which is moving down a microtubule. That motor protein, it's just a protein. It's walking just based on simple chemical interactions with that microtubule. So lysosomes are sort of a special type of vacuole, or vesicle. Lysosomes are an enzyme, have enzymes in them. These enzymes can be used to break things down. Lysosomes remove junk. What they really do is they're recycling things that the body doesn't need anymore, so you can use those basic building blocks over again. Because remember, we have just 4 real basic building blocks for living things. So lysosomes are used in the breakdown of organelles that have outlived their usefulness. Lysosomes can break down bacteria if a white blood cell gobbles it up by phagocytosis. So they are going to be the cleanup crew, breaking things down back to its basic components of amino acids or nucleotides or simple sugars will be done by a lysosome. Supporting the inside of the cell is a cytoskeleton. Just like we have a skeleton that is supporting us as a framework, your cells have a cytoskeleton. Now, this is made of protein. It's not made of calcified tissue. So there are a few different fibers of the cytoskeleton. So there are microfilaments, are thread-like structures made of a protein called actin. So the structure that we saw the motor protein walking on, this is going to be a microtubule. It is part of the cytoskeleton. We can see more of it in the background. This cytoskeleton can be built and taken apart with ease. It's not a permanent structure. So microtubules are hollow structures made of proteins called tubulins. They play a critical role in maintaining cell shape. So these microtubules, they're made of these 2 tubulin proteins. They are hollow down the center. That's what we saw the little guy walking his way down. So centrioles are located near the nucleus. They help to organize during cell division. So centrioles are not found in plant cells. There's still things I don't understand about that, and that I think science doesn't quite understand about that. But these centrioles are made out of microtubules in groups. Microtubules also help to build structures of cilia and flagella. So flagella are these long tail-like projections that allow single-celled things to swim. The only human cell with a flagella is the sperm cell. Bacteria can have 1 or multiple flagella. Cilia are similar to flagella, but they tend to be smaller and shorter. So bacteria can move around with a whole bunch of cilia. We have cilia in our respiratory tract, which helps sweep crud out of our lungs so we can cough it out of our bodies. Ribosomes are little structures. So these are structures made of RNA and protein. Ribosomes produce protein. So we are going to spend half a chapter basically talking about what the ribosome does. The ribosome does what we call translation. It takes mRNA and it produces protein from those mRNA instructions. Now we're going to get to the endoplasmic reticulum. So eukaryotes contain an internal membrane system known as the endoplasmic reticulum, or the ER for short. So there are two types of endoplasmic reticulum. There is the rough endoplasmic reticulum, which is so named because it has all these little bumps on it. Those bumps are ribosomes that we would see under electron microscope. Then there's the smooth ER, which is so named because it doesn't have the bumps, it doesn't have any ribosomes on the smooth portion. So the rough endoplasmic reticulum is involved in the synthesis of proteins. This should make sense because ribosomes are used to synthesize proteins, and the rough ER is covered with these ribosomes. The smooth endoplasmic reticulum does not have any ribosomes attached. The smooth endoplasmic reticulum helps to synthesize lipids, and it detoxifies some drugs. Proteins that are produced in the rough endoplasmic reticulum can then be transported to the Golgi apparatus. The Golgi apparatus is sort of the Amazon Prime of the cell. It receives packages, it modifies them a little bit, and then it sends them off to other places. It's a series of flattened membranes, but it's going to get vesicles coming in and send vesicles going out. So the Golgi apparatus modifies, sorts, and packages proteins. From the Golgi apparatus, proteins are shipped to their final destination inside or outside of the cell. So this is going to bring us to the last two organelles that we're going to talk about today. The last two organelles we're going to talk about today are the mitochondria, the powerhouse of the cell, and the chloroplast. So the mitochondria and chloroplast are two of the most important organelles. They are both involved in energy transfer. Obviously, all living things need a source of energy. Most organisms get their energy from food molecules, which ultimately come from the sun. So chloroplasts and mitochondria are both involved in energy conversion processes within the cell. Animals have mitochondria, plants have mitochondria, and they have chloroplasts. So chloroplasts are the biological equivalent of solar panels. They do the reaction of photosynthesis. They take in carbon dioxide and water, and they produce sugars and oxygen. Those sugars are storing the energy that is used by life. So plants are green due to their chloroplasts. So inside of the chloroplast, there is a pigment called chlorophyll. Chlorophyll is the green pigment of the plant that we find within the chloroplast. Mitochondria are the powerhouse of the cells. They convert the chemical energy stored in food into this substance, ATP. ATP is what your body uses for immediate energy. You have used billions of ATP molecules since you started to listen to this video. So mitochondria have an inner and an outer membrane. The inner membrane is folded up a whole bunch. We are going to have a whole chapter on the chloroplast. We're going to have a whole chapter on the mitochondria because the functioning of these two organelles is critical to life. So in humans, you get all of your mitochondria from your mother. The sperm cell does not transfer any mitochondria. Interestingly, the mitochondria have their own DNA. So chloroplasts and mitochondria have their own genetic material. We have endosymbiotic theory that states that chloroplasts and mitochondria have descended from independent microorganisms. This theory was first proposed by Dr. Lynn Margulis. It is now widely accepted and highly supported because it is the only thing that explains why mitochondria have their own DNA, why chloroplasts have their own DNA, why they replicate themselves and are not controlled by our nucleus. So the endosymbiotic theory states that there was some sort of ancestral primitive cell. It took in mitochondria, it took in chloroplasts, and instead of just devouring these organisms, instead these organisms began to live inside and they became symbiotes with us. And today we rely on these mitochondria and we will rely on these chloroplasts for basic life functions. So that is going to complete the notes for today. Tomorrow you are going to get a bookwork assignment. I'm going to be posting vocab that will be due tomorrow as well. Thank you. Have a good day.